Η ιατρική για να προάγει την υγεία πρέπει να ερευνήσει την αρρώστια, όπως η μουσική για να δημιουργήσει την αρμονία πρέπει να ερευνήσει την δυσαρμονία. Τα αδέφη ένας μέγας αρχαίος Έλληνας ιστορικός, ο Πλούταρχος, ο οποίος έζησε 1900 χρόνια πριν. Αυτή η φράση περιγράφει επακριβώς το θέμα της επόμενης ομιλίας εδώ στο Athens Science Festival. Ο Μανόλης Κέλης, ή Καμβισέλης όπως είναι το πλήρες όνομά του, το όνομα της οικογένειάς του που μετανάστευσε στην Αμερική πριν από σχεδόν 30 χρόνια, είναι καθηγητής επιστήμης των υπολογιστών του MIT και μέλος του εργαστηρίου επιστήμης υπολογιστών και τεχνητής νοημοσύνης. Είναι επίσης ένας επιστήμονας που έχει συνεισφέρει σημαντικά στην κατανόηση του ανθρώπινου γονιδιώματος και στην πρόοδο της υπολογιστικής βιολογίας. Για να αποκωδικοποιήσει τους μηχανισμούς που προκαλούν την ασθένεια, η ερευνητική του ομάδα μελετά τις γενετικές διαφορές μεταξύ των ανθρώπων που οδηγούν σε διαφορετικές νόσους, σε κυταρικό και μοριακό επίπεδο. Παρατηρεί, αναλύει, συγκρίνει κύτταρα από δείγματα υγιών και ασθενών, δημιουργεί εκατομμύρια χάρτες γονιδιωματικής δραστηριότητας και προσπαθεί να εντοπίσει το λάθος. Ποιες διαφορές, ποια γενετικά κυκλώματα, ποιες διαδρομές οδηγούν στην νόσο του Alzheimer, ποιες στη σχιζοφρένεια, στις καρδιακές παθήσεις, στην παχυσαρκία, στον καρκίνο. Τέλος, προσπαθεί να αναπτύξει τα εργαλεία για να τροποποιήσει αυτά τα κυκλώματα, είτε για να αποτρέψει την έκφραση της νόσου, είτε για να την θεραπεύσει. Στην ομιλία του με τίτλο «Σπάζοντας τους κώδικες της ανθρώπινης ασθένειας», ο κορυφαίος Έλληνας επιστήμονας θα παρουσιάσει τις ευκαιρίες σχεδιασμού νέων μεθόδων διάγνωσης για την αντιμετώπιση ανίατων μέχρι σήμερα ασθενειών, για περισσότερη υγεία, για μεγαλύτερη αρμονία. Τη συζήτηση συντονίζει η Ζωή Κούρνια, βιοχημικός, ερευνήτρια στο Ίδρυμα Ιατροβιολογικών Ερευνών της Ακαδημίας Αθηνών.
Γεια σας, καλησπέρα. Είμαι η Ζωή Κούρνια, είμαστε ζωντανά από το Ίδρυμα Ιατροβιολογικών Ερευνών της Ακαδημίας Αθηνών και έχουμε την μεγάλη χαρά και την τιμή να συνδεθούμε με τον καθηγητή Μανώλη Κέλλη και θα συνδεθούμε live με τη Βοστόνη αυτή τη στιγμή. Η ομιλία θα είναι στα αγγλικά και θα συνεχίσουμε με τις ερωτήσεις στα ελληνικά. Οπότε θα προλογίσω τον ομιλητή μας στα αγγλικά. Professor Manolis Kellis is a world-recognized leader in the field of precision medicine, disease genomics, digital health, machine learning, and computational biology. He has made important discoveries for Alzheimer's disease, obesity, schizophrenia, cardiac disorders, cancer, and immune disorders. And his research has broad implications for the study of all human disease. So he has received all his titles from the Massachusetts Institute of Technology, where he is also currently a professor. He has received a long list of awards, just to name a few. The U.S. Presidential Early Career Award in Science and Engineering, by President Obama, the Mandel Medal for Outstanding Achievements in Science, the NSF Career Award, the Alfred P. Sloan Fellowship, and so many others. He is very prolific in his scientific publications, highly cited. He has lived in Greece and France before moving to the US. And right now, it's our great pleasure and honor to have him in the Athens Science Festival where he will speak. So thank you very much, Professor Kellis.
Ευχαριστώ πάρα πολύ, Ζωή, και ευχαριστώ σε όλο το φεστιβάλ για την πολύ όμορφη οργάνωση που έχετε κάνει και για την πολύ όμορφη παρουσίαση από τόσο πολλούς επιστήμονες, μουσικούς και πολλούς ηγέτες σκέψεων πραγματικά στην Ελλάδα. I will actually switch to English for my talk, and I will share my work on dissecting the circuitry of human disease at single cell resolution. What we are trying to understand is how differences in the DNA of individuals is actually leading to disease. And here I'm actually showing 3 of my own differences in my own DNA. So these 3 differences are in fact increasing my risk for age-related macular degeneration, a form of blindness that can cause me to lose the central part of my vision as I grow older. How do we know this? We know this by systematically associating millions of single nucleotide differences of a single letter across millions of nucleotides in the human genome to recognize regions that are either protective in green or risk increasing in red associated with disease. What my lab tries to do is understand how we go from this individual difference to ultimately the disease pathogenesis. Namely, we're trying to bridge the gap between individual nucleotide letter differences and disease. And that gap is a very large gap because these differences don't act directly on disease. What they do instead is they affect specific tissues and specific cell types within these tissues. Within them, they lead to epigenetic differences, which are most of the time differences in the circuitry rather than the genes directly. And these lead to very often gene expression changes and very often intermediate endophenotypes that we can measure to understand the path between genetic variation and ultimately the manifestation of disease. But of course, that path is affected not just by genetics, but also by the environment and by the disease state itself. So what we're doing in my lab is systematically integrating large-scale data sets in order to do that. And the reason for that is that the promise of genetics has been to systematically understand these genetic differences and translate them into new disease mechanisms, translate them into new target gene predictions, translate them into new therapeutics, and ultimately enable personalized and precision medicine. Here I'm showing you one such example in the case of obesity, body mass index. I am, again, a double risk homozygous for obesity. And both my mom and my dad gave me the worst possible copy of this particular gene. And this particular region leads to the strongest genetic association with obesity. It leads to an extra 7 pounds of weight to the average adult, which is an enormous phenotypic consequence. And when you look within this region to try to understand the mechanism of this disease, what you find is that there isn't just one, but there are 89 common variants. These are common single nucleotide alterations that are co-inherited. If I have this one, I also have all of the other ones. What does that mean? That means that we don't really know which one is actually responsible for the disease. So we can actually start asking which ones of those perturbs the protein for this FTO gene that was promptly renamed fat and obesity associated when it was discovered to be the strongest genetic association with obesity. But the challenge, of course, is that none of them perturbs the protein. Only 1% of the human genome codes for proteins. The other 99% is much more difficult to comprehend. And that's what my lab focuses on. Why? Because in 90% of cases, these disease-associated regions do not perturb the protein. That means we don't know the target gene. We don't know the causal variant. We don't know the cell type where they act. And therefore, we don't know the relevant pathways, and we don't know the mechanism. And that's what my lab tries to do systematically across 120,000 disease loci. So how do we do that? We start with genetic variation. We start with the amazing results of human genetics over the last many, many decades to systematically understand what are the genes and what are the single nucleotide letters that are associated with disease across both common variants, which are shared across the human population, but also rare variants that happen in families and in isolated populations. We then try to make sense of these variants by bridging that gap between genetic variation and disease. How do we do that? By systematically profiling the molecular circuitry of every single one of our cells. By profiling the RNA transcription that basically tells us which genes are expressed, which proteins are produced in the context of disease and in the context of healthy samples. And number two, we profile the circuitry. And for this, we use the epigenome. Epi, from Greek, of course, on top of the genome. So what is epigenomics? Epigenomics is the collection of all of the post-transcriptional and post-translational modifications that DNA, RNA, and histones can undergo. The DNA that we all know and love is not in this beautiful double helix form, swimming around naked inside our cells. It is wrapped around these histone proteins, which can undergo post-translational modifications. What does that mean? That means that every cell needs to remember its identity. My brain cells and my lung cells and my liver cells and the eyelids and my cornea and so on and so forth, every single one of them have very different biological functions, and yet they all have exactly the same DNA. The way that they achieve this extreme diversity of functions across trillions of cells in our body is by, quote unquote, remembering the state of every one of these genes and every one of these control regions. Namely, every gene has upstream control elements that will govern whether that gene is turned on or off. And these control elements are maintained in an epigenetic state that distinguishes liver cells from brain cells and so on and so forth. So what we're doing in my lab is systematically measuring that epigenetic state to understand which control regions are turned on and turned off in every single cell type of the human body. We're using the accessibility of DNA that basically tells us if it's, quote unquote, open for business, if the different protein regulators that need to turn on the genes can bind there or not, and the modifications of histone proteins that recruit particular regulators that change the conformation of these regions to be more accessible, more protected, and so on and so forth, which ultimately leads to the RNA expression. So the first step that we do is we measure both RNA, the output, if you wish, of the circuitry, and epigenome, the input, if you wish, of the circuitry, in both healthy and disease samples to try to understand how they work together, how they differ in response to genetic variation, and how they differ between diseased individuals and healthy individuals, whether those differences are due to genetics or whether those differences are due to the environment or to the disease itself. Once we've gathered thousands of samples across thousands of individuals, we then integrate all these data sets computationally. We develop Bayesian methods, deep learning methods, inference methods, causality methods, mediation methods, to try to understand how genetic differences ultimately lead to the circuitry being altered and ultimately the genes being dysregulated, and ultimately the pathways associated with disease being changed and leading to disease. So we predict driver genes, regions, and cell types using this computational machine learning, AI, large-scale data integration in order to predict this circuitry. And then we go and manipulate this circuitry using mouse models and using human cell cultures to validate the predictions, to show that indeed, if we change that nucleotide, or if we change this epigenomic region, or if we change the expression of that gene, how does the pathway change? And how does ultimately the cellular signature and the organismal signature of disease change? We, of course, disseminate the results and start all over again. The first step, as I mentioned, is large-scale data gathering. That means that we have collaborated with clinicians and physicians across the world, across many, many different institutions, to collect samples, very often post-mortem, of the human brain for Alzheimer's disease, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar disorder, Down syndrome, autism, depression, suicide, post-traumatic stress disorder for the brain, but also live samples from living individuals in the case of obesity, exercise, heart disorders, cancer. We are basically taking samples from living individuals to study a very diverse range of disorders. We're looking at dozens of traits, each across several dozens of individuals, totaling thousands of individuals across our samples. And then we're looking at every single cell type using single cell profiling. So instead of taking the whole brain, we're basically saying, no, we're going to profile every single individual cell across thousands of cells from the brain of disease and healthy individuals, because every single cell type of the brain has very different functions and very different activity. So if we mush it all together, it's indecipherable to understand how these individual nucleotide variants act. But if we have single cell resolution, then we can systematically understand those differences. So neurons, oligodendrocytes, astrocytes, microglia in the human brain, but dozens of immune and many other cell types across many different disorders allow us to now start understanding how these genetic variants are acting, what gene regulatory elements they're affecting, and ultimately, how are they affecting disease. We then profile for those in single cell resolution, both the RNA expression and the DNA accessibility, the epigenomics. And we do that across many different brain regions and across heart, muscle, adipose, and many other organs. We then integrate all these data sets. So many of my students are computational students. So what they bring is the expertise to integrate these massive data sets using Bayesian methods to infer causality variables using the prior of epigenomic annotations, the posterior evidence for something being associated with disease, and ultimately a causal indicator that integrates this prior and the likelihood from the genetic association to predict which genetic variants are in fact drivers of the disease. We've developed both Bayesian and non-Bayesian methods for causality inference and for mediation analysis to understand what are the intermediate variables that mediate the effect of genetic variation on disease. And we're using imputation, we're using many other methodologies for understanding that path to disease. We've developed deep learning methodologies. Just like deep learning has now allowed us to understand images and develop intermediate representations of those images by thinking about the concept of a wheel or the concept of a face, of a nose, of an ear, through the edges, through layers of abstraction, we are now developing the same abstraction layers for the DNA, starting from the individual DNA words at the lowest level, combined together into sentences and phrases and paragraphs together to lead to cellular function, so that we can then understand how individual nucleotide differences are changing the words, the sentences, and the meaning of these paragraphs to ultimately cause disease. And we're also developing methods for predicting which cell types are causal using heritability partitioning and statistical genetic approaches to understand what are the relevant epigenomic annotations underlying immune disorders, psychiatric disorders, pointing to the central nervous system or T cells and B cells, and so on and so forth. After we integrate all these data sets, the next step is to infer these circuits and manipulate these circuits. To give you an example, a few years ago, we published a paper that showed the circuitry underlying this genetic variant that I showed you on the first slide that predisposes me to obesity. We basically found out that out of these 89 common variants that I had mentioned, we could pinpoint this entire genetic association to only one variant, this T to C alteration that changes the letter T into the letter C, and that disrupts this AT-rich interacting domain, this AT-rich binding protein that binds AT-rich motifs, but then when the T in the middle is changed to a C, can no longer bind. We found out that we can trace this difference to both its upstream regulator and to its downstream target genes. How? By understanding through large-scale EQTL datasets, expression quantitative trait locus datasets. These are databases where we can look for how genetic differences correlate with epigenetic differences and transcriptional differences in control individuals. That's the beauty of it. The fact that because these common genetic variants are shared between so many different people, we can in fact start understanding them systematically. So when I say we want to understand 120,000 genetic loci in the genome, it is actually a feasible task because we can actually profile all of those across millions of samples, across thousands of individuals. So what we found is that this genetic alteration is in fact leading to a change in expression in two genes, not at all the FTO locus that this variant is found in, but instead two genes that are found 600,000 nucleotides away and 1 million nucleotides away. That basically means that instead of where the genetic variant is, we have to look far, far away in the genome, several genes over, to find the true targets of these genetic variations. So we now have the circuitry. We have the upstream regulator, we have the single nucleotide variant, we have the downstream target genes, and we have the control region, this 10,000 nucleotide long super enhancer, this enhancer, which is a master control circuitry, which is acting early in the differentiation of white adipocytes and beige adipocytes. We all think of fat as our enemy. Oh, the more fat you have, the more sick you are. And that's true for white fat. White fat is what stores all of these excess lipids inside our body. And in fact, if we didn't have fat, all of these lipids would be throughout our blood. So even our white fat is our friend because it protects all our other cells from the damage that these lipids would be carrying out. But white fat stores energy for a rainy day. For food scarcity periods, we are able to now save this extra energy to use it when we don't have as much food. And that's the variant that I have inherited. I store all of my excess calories into my white adipocytes. But what this circuit does is that it chooses whether it should burn these calories in a process known as thermogenesis to basically get rid of these calories in the form of heat by depolarizing the mitochondrial membrane and then causing the loss of this proton gradient and ultimately the loss of energy as heat. So what happens for those of us who have inherited this obesity-inducing mutation is that the upstream regulator can no longer bind here. It can no longer repress this enhancer. This enhancer becomes overactivated. It overactivates the downstream target genes, which then shut off thermogenesis and turn on lipogenesis, causing us to ultimately develop obesity if we eat too much, if we don't exercise strong enough. Why am I excited about this? I'm excited because, number one, of course, I understand my own circuitry of my own cells of why I gain weight more easily, number one. But number two, by revealing the circuitry, we can actually manipulate the circuitry. And what we have shown is that we can actually go in and intervene. We can change the upstream regulator. We can change the downstream target genes. And we can even use CRISPR-Cas9 genome editing to change that single nucleotide variant and then understand how these are in fact allowing us to switch back and forth between lean and obese phenotypes. when we take primary cells from individuals like me that carry the risk allele and that are unable to thermogenize, and we edit a single letter from these primary adipocytes in a dish, from a C, which is disrupting this motif, to a T, which restores the motif and restores the binding and restores the expression, the repression of the enhancer, and shuts off these genes, what we see is that, in fact, the process of thermogenesis is completely restored. The cells from these obesity-prone individuals are, in fact, able to now burn calories by changing 1 out of 3 billion letters of the human DNA. Going into mice, we can now start asking, beyond the cellular consequences, what are the organismal consequences of repressing these two genes whose overexpression leads to obesity? And what we find is that repressing these genes, even in normal mice, causes these mice to completely deplete all of their fat stores. They simply burn away all of their fat. They're stuck in this thermogenesis position. They start out leaner, and when you feed them fat, normally mice gain weight. These mice are unable to gain weight. They are just simply stuck, burning every calorie in their body when they're awake and also when they're sleeping. So that's the promise of understanding circuitry. We don't simply want to say, oh, sorry, bad news, you have a predisposition to disease, which is already very informative because then I can act upon it. I can change my diet, I can change my exercise pattern, and so on and so forth. But we can actually go and say, well, pharmaceutically, we can now start intervening and changing the metabolism of our cells, in addition to changing our behavioral patterns, in order to help our body combat different diseases. So that's what my lab does systematically. We basically, number one, develop these maps of the epigenome across hundreds of tissues in order to understand the circuitry of disease. Number two, we try to dissect these very complex disorders into simpler parts. For example, if we take coronary artery disease, it's an extremely polyfactorial disease. It has many, many different facets. It acts in the liver, in the coronary artery, in the thyroid, adipose tissue, and so on and so forth. And in every one of these tissues, it affects different pathways. By having that epigenomic circuitry, we can actually start understanding its components, dissecting them individually, and then going back into individual disease loci to start acting which tissue do they act in, what target gene do they act through, and how can we manipulate them in order to reverse the disease phenotypes. And we've now done this systematically across 30,000 different loci in a paper that we just published in Nature just a few weeks ago. Beyond the tissue level, we've also been studying all of these at the single cell resolution. So in a paper that we published in Nature last year, we basically showed that we can now start understanding Alzheimer's disease in single cell resolution, understand thousands of cells across dozens of individuals, both phenotypically normal prior to Alzheimer's, early, and then late and severe Alzheimer's. And what we're finding is that every single major cell type shows dramatic alterations associated with Alzheimer's disease. And we also find that female individuals show a much higher preponderance of these disease states, suggesting that they in fact have a higher risk. We're able to understand what are the differences that are happening early in the disease versus late in the disease, and what are the sex-specific differences that are happening. We're finding that there are thousands of genes that are differentially expressed between men and women prior to Alzheimer's, but thousands more that are differentially expressed when Alzheimer's actually strikes. That allows us to now start understanding disease in a sex-specific, stage-specific, cell type-specific way. And in particular, we're finding that our methods predict that women would have more myelination loss. This is the coating of our axons, that protects them and sort of helps the signals in our brain travel more easily. And indeed, from the single cell data, we predicted that there would be a difference. And when we went and checked, we found that indeed, women show a lot more white matter loss in their brain during Alzheimer's than men. For schizophrenia, we're able to again go in at single cell resolution and understand what are the genes that are changing in expression in schizophrenic brains that are associated with predisposition for schizophrenia. So we can now go across dozens of different genetic loci and predict what is the candidate target gene. Is it positively or negatively associated with schizophrenia, leading to very different therapeutic strategies for repressing or overexpressing the gene, and exactly what cell type in the brain should be altered and should be targeted to combat psychiatric disorders. We're also able to distinguish a different path to schizophrenia, where individuals that show normal gene expression patterns have one cellular state that appears to disrupt the whole process, even though globally they don't show these global gene expression patterns, suggesting perhaps another form of treatment for the approximately 1/10 of individuals that do not show these typical gene expression alterations. We're able to use these data to now start predicting who are the master regulators of schizophrenia and where do they act. And what we're finding is that they act both early in development and also in the adult brain, suggesting a link between neurodevelopment and then synaptic organization, which are both implicated in schizophrenia. Beyond just the prefrontal cortex that I showed you so far, we're able to look at many different regions of the brain, including the hippocampus, the entorhinal cortex, which are some of the earliest changes that are happening in Alzheimer's disease. And what we're finding is extreme diversity in the neuronal cell types that are acting in each of these regions. We're finding new genetic markers for 30 or more excitatory neurons and 23 inhibitory neuron subtypes in all of these cell types. And we're finding that not only are the neurons different between different regions, but also that the glial cells, the astrocytes, the oligodendrocytes, the microglia, are in fact dramatically different between cortical and subcortical regions of the brain. And we're also able to build a time course of disease progression by taking these millions of individual cells across these 7 different brain regions and across 48 individuals each, and then tracing which cells are early in the disease progression using computational methods that allow us to predict Alzheimer's, and late in the disease progression, enabling us to study how every gene turns on at different stages of disease progression to understand how to intervene early in the disease. Beyond the single cell resolution, we're able to understand the spatial organization of these brain structures. If you look at the hippocampal formation, you see CA1, 2, 3, the dentate gyrus, the subiculum, and the entorhinal cortex here have very precise organization in 3 dimensions. And what we're starting to do now is look at how the single cell data sets allow us to predict the 3-dimensional positioning of these cells by taking coupled data with spatial transcriptomics and single cell data, and then using, number 1, a deconvolution approach where we can take the spatial data and deconvolve it into the single cell data set that we can infer from what I like to call these mini bulk profile. And we're also developing these deep learning methodologies to predict the spatial organization and the spatial localization of a cell using the single cell expression patterns. We're able to discover many different subtypes of microglia. Some are synaptic and some are inflammatory. And it turns out that the inflammatory microglia playing roles in Alzheimer's, but the synaptic microglia play roles in schizophrenia. We're also able to study the vasculature of the brain and understand what are the cells that control the blood-brain barrier, that control the influx of nutrients and other chemicals from the blood supply to our brain, and how these are in fact underlying both neurodegenerative disorders and psychiatric disorders. We're able to look at not just the RNA changes, but also the DNA accessibility changes to start pinpointing the regions of gene regulation that are responsible for these genetic variants and how they act. And we're able to pinpoint these very subtle changes that are happening in specific cell types in the brain and the accessibility of individual regulatory motifs and how that's changing the context of disease. And what we're finding quite dramatically is that even though excitatory neurons and inhibitory neurons appear to underlie psychiatric disorders, it is instead the microglia, the immune cells of the brain, that appear to be underlying the genetic risk to Alzheimer's disease, suggesting that, in fact, immune dysregulation might be at the basis of Alzheimer's disease much earlier than all of the amyloid and tau pathology that we see later in the disease progression. We're expanding this across many different tissues of the body, not just looking at the brain, but looking at the liver, the heart, the blood, the intestine, the white and brown fat, and the muscle. And we're looking at all of that in the context of both human and mouse to understand how exercise impacts all of our tissues to basically lead to increased health. And we're discovering dozens of different subtypes of cells and how they're changing with both exercise and obesity. And what we're finding is quite dramatic. Number one, the adipocyte stem cells, the early stage of differentiation of our fat cells, where they decide to be fat storing or fat burning, appear to be showing the strongest changes, along with the immune cells, the gene regulatory cells that play both immune functions, but also control functions, and that control very often the communication patterns between different cells of our body. So all of our tissues are affected by exercise. How is that possible? Well, the circulating blood cells, the hormones that are being secreted by the exercise muscle, all of these things are permeating through the body through receptors and ligands that are interacting, that are sending these messages from one cell type to the other within the same tissue, and from one tissue to the other across the body. So all of these things together, we can now start systematically understanding, and again, start asking, how do we intervene to change both the state of early adipocyte differentiation and the state of the circulating blood, giving us a handle for intervening and repeating this beneficial effect of exercise across all our tissues. In the context of cancer, we're looking at the progression of cancer across immunotherapy, the changes that are happening in different lineages that are coexisting within the body of these individuals in a live progression of disease with biopsies that are getting sampled as every patient goes into the surgery. The surgeon basically sends us the samples, and within the same day, we profile them to understand how the cancer is progressing and whether the drugs are in fact working or not. And we're building predictive models that allow us to predict which therapy will work the best for each individual. And we're also integrating electronic health records with the genetic information to start understanding how these global comorbidity patterns across disease can be inferred from the doctor notes using natural language processing, the prescriptions, the billing codes, the lab tests that are prescribed, and the results of these tests in a hierarchical model that allows to integrate all these data in a single framework. And then lastly, because my lab is making thousands of hypotheses across the genome, we have also developed computational methods that allow us to test millions of hypotheses in a single experiment. This computational student in my lab developed this new experimental method, which he then tested and showed that he can actually measure the activity of 7 million different enhancers in a single experiment by cutting them out of the DNA, inserting them into thousands of plasmids, and then testing each of those in millions of cells to be able to localize precisely the nucleotides that are responsible for epigenomic activity and for gene regulatory activity. And we've now developed programmable constructs using both genome editing, but also using dead Cas9, a variant of the CRISPR-Cas9 system that simply recruits activators or repressors to particular enhancers to be able to turn on and turn off individual gene regulatory elements in different cell types. We apply those in induced pluripotent stem cells, which we then differentiate into neuroprogenitor cells, neurons, astrocytes, oligodendrocytes, and microglia, to understand how these circuit manipulations are acting in each one of our cells. These are modular, and these allow us to now start testing the upstream enhancers individually. And in collaboration with Andreas Fenning, a former postdoc in my lab, who's now a professor at CMU, we're now testing thousands of these genetic manipulations in living brains of mice to understand how these act, not just in a dish, but also in the context of the whole animal. Lastly, our lab has been contributing to the fight against COVID by systematically understanding the genome of the SARS-CoV-2 virus, being able to compare multiple related species, to revise the gene annotation, which turns out to have been wrong in multiple places. This gene, for example, is an RNA gene, is a non-coding RNA. This is not a protein. And we were able to also discover a new gene that was previously overlooked because it overlaps another gene. It's a gene that's encoded within another gene that now we believe has particular functions specific to SARS-CoV-2. And we're able to understand the mutational patterns of each of these genes across evolutionary time and also within the current pandemic by integrating data across thousands of sites that have been sampling coronavirus samples and DNA across the entire world to understand what is the rate at which different genes evolve within the current pandemic. And we're finding that the nucleocapsid protein is appearing to be evolving much more rapidly within the current pandemic than we would have expected. And in fact, these mutations appear to be particularly affecting a B-cell epitope and perhaps causing immune evasion for the virus. By contrast, the spike protein appears to be evolving significantly less rapidly. And we're also able to study the mutations that you have all heard about in the news in the context of their evolutionary history. And in particular, we're finding, for example, that this D614 gene mutation that appeared very early in the pandemic and that has led to increased transmission and has become the dominant strain across the world is in fact a human-specific change that has never been seen in any of the relatives of the current SARS-CoV-2 virus in any of their bat hosts, suggesting it might actually be a specific human host adaptation. I want to say that this is the work of many, and we're collaborating with many within MIT and across the larger community. This is something that I think Athens and Greece should replicate as much as possible, with closer ties between industry and academia. And there's many professors across computer science, biology, engineering that are involved in many different aspects of this endeavor. And lastly, many of our students have gone off to their own faculty positions, and we have many new grants, so we're looking forward to working with you guys. So if you have students and postdocs and bright people who are excited, computer savvy, or experiment savvy, and who want to work on all these topics, we are always hiring. So search for compbio.mit.edu. And this is just a quick summary. We basically integrate genetics, epigenomics at single cell resolution, computational integration, and then systematic manipulation in both cell cultures and mouse models. So I'll stop there and I'll be delighted to take any questions.
Ευχαριστούμε πάρα πολύ για αυτή την φανταστική ομιλία με όλα αυτά τα φανταστικά prospects που μπορεί να περιμένουμε από την τεχνολογία πολύ σύντομα με βάση τη δική σας έρευνα. Ήθελα να κάνω μία ερώτηση, γιατί ήταν πάρα πολύ ενδιαφέρον πώς συνδέσατε όλη την ομιλία με το δικό σας γενετικό κώδικα, το οποίο ήταν πολύ ενδιαφέρον να το ακούσουμε. Οπότε η ερώτηση είναι, αν κάποιος έχει πρόσβαση στο γενετικό κώδικα, αλλά δεν είναι ο Μανώλης Κέλλης και δεν μπορεί να την αποκωδικοποιήσει όπως εσείς, αλλά παρόλα αυτά μπορεί να απευθυνθεί σε ειδικούς και να ανακαλύψει ότι έχει πιθανότητα να πάσχει από κάποιο σύνδρομο. Στην περίπτωση της παχυσαρκίας καταλαβαίνω ότι μπορούμε να κάνουμε πράγματα, αλλά στην περίπτωση του macular degeneration, το οποίο νομίζω είναι εκφυλισμός της οχράς κοιλίδας, για το οποίο δεν υπάρχει ασθένεια, πόσο χρήσιμη θα είναι αυτή η πληροφορία στον απλό πολίτη?
Ομολογώ ότι ένας πολύ καλός μου φίλος έχει αυτή τη στιγμή χτίσει μια εταιρεία, την οποία τη λένε Alcus, η οποία βοηθάει όχι μόνο στο AMD, αλλά και στην Stargardt disease, που είναι μια μονογεννητική ασθένεια που ξεκινάει πολύ, πολύ πιο νωρίς και τα παιδιά χάνουν την όρασή τους από πολύ, πολύ μικρά. Και έχει δείξει αυτή τη στιγμή σε clinical trials ότι όντως μπορεί να σταματήσει εντελώς αυτό το χάσιμο της όρασης των παιδιών. Οπότε υπάρχει ελπίδα και υπάρχουν πολλές ασθένειες στις οποίες ξεκινάνε τώρα καινούργια φάρμακα. Τώρα, υπάρχουν όμως και πολλές μελέτες που δείχνουν ότι μπορείς να αποφεύγεις τον ήλιο, μπορείς να φοράς μαύρα γυαλιά, μπορείς να τρως πιο πολλά καρότα, μπορείς να κάνεις πιο πολλή άσκηση. Νομίζω ότι μπορούμε να κάνουμε διάφορες αλλαγές στην περιφορά μας, στην καθημερινή μας ζωή, που μας βοηθάνε γενικώς με πιο υγιεινή διατροφή, με πιο πολλή άσκηση, με πιο πολλή ξεκούραση, ύπνο. Όλα αυτά βοηθάνε σε όλες τις ασθένειες. Όσο για τις ασθένειες τις οποίες δεν υπάρχουν αυτή τη στιγμή φάρμακα, το μόνο που μπορώ να πω είναι υποστηρίξτε την επιστήμη, γιατί όπως είδαμε σε αυτή την πανδημία, μπορούμε πολύ πολύ να αλλάξουμε την κατάσταση του πλανήτη ολόκληρου, άμα οι επιστήμονες έρθουν όλοι μαζί και συνεργαστούν. Οπότε ελπίζουμε όχι μόνο για τις ασθένειες που υπάρχουν τώρα φάρμακα, αλλά και για πολλές άλλες ασθένειες να υπάρχουν λύσεις πιο σύντομα.
Όντως αυτό είναι πολύ σημαντικό και νομίζω ότι είδαμε μια γενικότερη στροφή της κοινωνίας προς την επιστήμη μετά και κατά τη διάρκεια μάλλον της πανδημίας. Ελπίζω να συνεχιστεί και μετά.
Θέλω να προσθέσω για την αρχή της ερώτησής σας που είπατε πώς μπορεί κάποιος να ξέρει για το ίδιο του το γονιδίωμα. Καταρχήν υπάρχουν πολλά service τα οποία κάνουν αυτό και για ολόκληρο το γονιδίωμα ή απλά για τους για τις πιο κοινές μεταλλάξεις και μεταλλαγές που υπάρχουν μεταξύ ανθρώπων. Υπάρχουν πολλά γεννητικά κέντρα, τα οποία μπορούν να σας βοηθήσουν να καταλάβετε αυτές τις διαφορές, γιατί ας πούμε, το να έχεις 8% αντί για 6% ρίσκο, τι σημαίνει για σένα? Πρέπει να αλλάξεις τον τρόπο που πρέπει να φοβάσαι κτλ. Οπότε το να καταλάβει κανείς τις στατιστικές, τις πιθανότητες και να καταλάβει και το πώς μπορούμε να αλλάξουμε τον τρόπο ζωής χωρίς να έχουμε το στρες, τα οποία μπορούν να μας κάνουν πιο πολύ κακό. Οπότε αυτό είναι το πρώτο. Και το δεύτερο είναι ότι πρέπει να ξέρουμε ότι το γονιδίωμα ναι μεν παίζει ρόλο και μερικές φορές μεγάλο στην ασθένεια, αλλά τις πιο πολλές φορές ο ρόλος του γονιδιώματος είναι πολύ μικρός σε σχέση με το περιβάλλον, σε σχέση με τη διατροφή, με την άσκηση και πολλά άλλα θέματα. Οπότε μην νομίζετε ότι είναι καταδίκη το να έχετε μια κάποια μετάλλαξη. Όπως βλέπετε, δεν είμαι εντελώς παχύσαρκος. Και αυτό είναι απλά θέμα συμπεριφοράς. Είναι θέμα άσκησης, είναι θέμα έλεγχου και είναι πώς ο κάθε ένας λαμβάνει όλα αυτά που μας έχουν δώσει γεννητικά και από το περιβάλλον μας και μπορούμε να κάνουμε ο κάθε ένας το καλύτερο που μπορούμε ως πρόσωπο και ως μέλος της κοινωνίας.
Άρα, γνωρίζοντας το γονιδίωμά μας, μπορούμε να προβούμε σε συμπεριφορικές αλλαγές που θα μας βοηθήσουν με την επιγενετική, ίσως να βρούμε κάποια άλλα μονοπάτια διάσωσης, στα rescue paths, ας πούμε, ή να μεθυλιωθούν κάποιες ιστώνες και να εμποδίσουν το onset της ασθένειας, κάπως έτσι.
Αλλά η έρευνα που κάνω εγώ στο δικό μου το group δεν είναι τόσο για τον κάθε έναν από εμάς, αλλά είναι για τους γιατρούς, για την φαρμακευτική βιοτεχνολογία, για τις εταιρείες που χτίζουν αυτά τα φάρμακα, για να τους βοηθήσουμε να ξέρουν τι φάρμακα να χτίσουν, για ποια κύτταρα και για ποιες ασθένειες. Δηλαδή αυτό που κάνουμε εμείς είναι κάτι το οποίο θα βοηθήσει σε 5, 10, 50 χρόνια για πάρα πολλές ασθένειες, αντί για 1 ή 2 ανθρώπους σήμερα.
Μάλιστα. Ευχαριστούμε. Ωραία. Έχουμε και μια ερώτηση από το κοινό. Νομίζω ότι το πρώτο σκέλος της ερώτησης, το οποίο είναι πώς η τεχνητή νοημοσύνη μπορεί να μας βοηθήσει να προβλέψουμε ασθένειες, μέσω των μεγάλων δεδομένων. Νομίζω ότι απαντήθηκε αρκετά, αλλά ίσως να θέλετε να κάνετε κάποιο σχόλιο από αυτού και αν οι κβαντικοί υπολογιστές θα φέρουν επανάσταση, να συνδέσουν το γονιδίωμά μας, το γενετικό μας κώδικα, με το φαινότυπο που έχει ο καθένα μας.
Λοιπόν, η τεχνητή νοημοσύνη παίζει τεράστιο ρόλο σε όλα τα κομμάτια για τα οποία μιλάω. Δηλαδή, δεν θα μπορούσαμε να κάνουμε τίποτα από αυτά που κάνουμε άμα δεν είχαμε στατιστικές, αλγόριθμους, τεχνητή νοημοσύνη και όλες αυτές τις μεθόδους που χρησιμοποιούμε για να μπορούμε να ενσωματίσουμε όλα αυτά τα δεδομένα. Αυτό είναι το πρώτο. Το δεύτερο είναι ότι αυτά τα quantum computing και όλα αυτά δεν νομίζω ότι θα παίξουν άμεσο ρόλο για ακόμα πολλές δεκαετίες στις δουλειές που κάνουμε. Δηλαδή, οι τεχνολογίες που χρησιμοποιούμε τώρα είναι κλασικά computer και πολλές φορές είναι τα graphical processing units, τα GPU, τα οποία βοηθάνε να χρησιμοποιήσουμε, ας πούμε, τα neural networks για deep learning και τέτοια θέματα. Οπότε υπάρχουν μερικές καινούργιες αρχιτεκτονικές υπολογιστών που μας βοηθάνε, αλλά δεν νομίζω ότι θα χρησιμοποιήσουμε αυτά τα quantum computing για αρκετό καιρό.
Όντως. Ήθελα να κάνω και άλλη μια ερώτηση σε σχέση με την αρχική ερώτηση, δηλαδή ότι μπορούμε να ξέρει ο καθένας μας το γενετικό του κώδικα. Εδώ ίσως προκύπτουν και κάποια θέματα βιοειθικής, γιατί αν ο καθένας μπορεί να κάνει αυτό το DNA τεστ στο παιδί του, το αγέννητο, στο έμβριο, πώς αυτό θα μπορούσε ενδεχομένως να επηρεάσει συμπεριφορές των ανθρώπων και το πώς οι άνθρωποι θα μπορούν να αποφασίζουν τι είδους παιδιά θέλουν να γεννήσουν.
Αυτό είναι μια τεράστια και πάρα πολύ σημαντική βιοειθική ερώτηση, η οποία χρειάζεται πραγματικά πολλές ώρες για να συζητήσουμε. Υπάρχουν με κάθε καινούργια τεχνολογία και τα καλά και τα κακά. Μπορούμε να αποφασίσουμε πώς θα τη χρησιμοποιήσουμε, αυτά τα δεδομένα, αυτή τη καινούργια γνώση, με τρόπους ηθικούς για να αποφύγουμε τις ασθένειες, όχι για να αλλάξουμε το χρώμα των μαλλιών μας και το χρώμα των ματιών μας και το πόσο ψηλός είμαστε κτλ. Δηλαδή, οι διαφορές μεταξύ ανθρώπων είναι αυτό που μας δίνουν στην ανθρωπότητα την ομορφιά της. Και το ότι υπάρχουν καλλιτέχνες, όπως έχουμε δει σε αυτό στο φεστιβάλ το ίδιο, από την jazz, στην ποίηση, στο κινηματογράφο, στην φυσική, σε όλες τις λογοτεχνικές και επιστημονικές κλάδους. Αυτό μας κάνει ανθρώπους. Τώρα να πούμε θα διαλέξουμε όλους τους ανθρώπους να είναι οι ίδιοι, δεν θέλουμε. Δηλαδή, όσο απίστευτος και να ήταν ο Αϊνστάιν, δεν χρειαζόμαστε 10 εκατομμύρια Αϊνστάιν στον πλανήτη μας. Χρειαζόμαστε ανθρώπους που είναι ο κάθε ένας διαφορετικός από τον άλλο, γιατί από εκεί έρχεται η γνώση, από εκεί έρχεται η έμπνευση, από εκεί έρχεται η ομορφότητα του πλανήτη μας. Για τις πολύ σημαντικές ασθένειες, βεβαίως, είναι κάτι το οποίο από τώρα πρέπει να αρχίσουμε να κοιτάμε για να βοηθήσουμε πρώτον να ξέρουν οι γονείς και να μπορούν οι γονείς ήδη να διαλέξουν. Δεύτερον, πώς μπορούμε να προσφέρουμε περίθαλψη για αυτές τις ασθένειες από την αρχή. Αντί να περιμένουμε να εμφανιστεί κάτι 60 χρονών ή 40 χρονών ή 20 χρονών όταν γίνει κάποιος, μπορούμε από την αρχή να πούμε: κοίτα, αυτό είναι μία πιθανότητα στο μέλλον σου. Ας πάρουμε τώρα τα βήματα που χρειάζεται για να μειώσουμε το ρίσκο για αυτή την ασθένεια. Βεβαίως, όχι να αρχίσουμε να σκοτώνουμε ανθρώπους οι οποίοι θα μπορούσαν να έχουν μια πανέμορφη ζωή και να συνεισφέρουν πάρα πολύ στην κοινωνία μας, αλλά να βοηθήσουμε αυτούς τους ανθρώπους να ζήσουν μια πολύ πιο όμορφη ζωή και μια πολύ πιο εύκολη ζωή.
Ωραία. Και θα πάρω και άλλη μια ερώτηση από το κοινό, η οποία μας ρωτάει αν με τη βοήθεια της τεχνητής νοημοσύνης θα μπορούσαμε μέσα στον αλγόριθμο να βάλουμε αυτές τις συμπεριφορικές αλλαγές που θα προτείναμε με βάση το γενετικό κώδικα και τα προβλήματα που βλέπουμε, και αν θα μπορούσαν αυτές μετά οι συμπεριφορικές αλλαγές που έχουμε βάλει στον κώδικα να μας πούνε ποια είναι η καλύτερη, πιο ενδεδειγμένη οδός, τρόπου ζωής για να αποφύγουμε το πρόβλημα το οποίο μας δίνει ο γενετικός μας κώδικας.
Συμφωνώ απολύτως Και αυτά τα δεδομένα μπορεί να έρχονται, όχι, δηλαδή αυτό που κάνω αυτή τη στιγμή με την πειθαρχία και την άσκηση, είναι ότι βλέπουμε πώς αλλάζουν όλα αυτά τα όργανα με την άσκηση. Και αυτό είναι κάτι το τρομακτικό, δηλαδή τρομερό, το πώς πραγματικά η άσκηση σταματάει όλα αυτά τα κακά που έρχονται με την πειθαρχία. Είναι κάτι το απίστευτο, δηλαδή όταν βλέπει κανείς τα δεδομένα, κάθε pathway έχει αλλάξει ακριβώς ανάποδα από την υπερτροφία. Αυτό είναι κάτι το οποίο μπορεί να μας βοηθήσει να καταλάβουμε ποιες συμπεριφορές, ποιες τροφές αλλάζουν ποια κυκλώματα μέσα στα κύτταρά μας και ανάλογα με το τι ασθένεια προβλέπουμε, ας πούμε, για τον κάθε ασθενή, μπορούμε να πούμε ειδού μερικές αλλαγές οι οποίες μπορούν να βοηθήσουν σε αυτές τις ασθένειες. Αλλά το πιο απλό που μπορώ να σας πω είναι κάντε άσκηση και τρώτε υγιεινά. Αυτό είναι κάτι το οποίο βοηθάει, ομολογώ, σε σχεδόν όλα.
Και αυτό προφανώς είναι μακροχρόνια η επίδρασή του, δηλαδή σε περίπτωση που πάρουμε το εμβόλιο ή πάρουμε κάποια φάρμακα, αυτά δεν μπορούν να αλλάξουν επιγενετικά το γονιδίωμά μας, γιατί είναι πολύ βραχυπρόθεσμα. Είναι σωστό αυτό?
Μιλάτε για εμβόλια και άμα μπορούν να αλλάξουν το γονιδίωμά μας. Οπότε πρέπει πολύ καθαρά να πω ότι τα εμβόλια αυτά δεν αλλάζουν το γονιδίωμά μας. Είναι κάτι το οποίο δεν έχει ελάχιστο ρίσκο σε σχέση με το τεράστιο ρίσκο το να μην εμβολιαστούμε. Οπότε ως επιστήμονας, ως πατέρας, ως άνθρωπος, μπορώ να σας πω, βοηθήστε την κοινωνία, πάρτε το εμβόλιό σας και ότι όλα αυτά τα οποία έχουμε ακούσει για τα ρίσκα είναι ελάχιστοι αριθμοί σε σχέση με εκατομμύρια δόσεις που έχουν πάρθει. Οπότε πραγματικά δεν πρέπει να νομίζουμε ότι υπάρχει ρίσκο με το εμβόλιο. Είναι κάτι το οποίο έχει ελεγχθεί πάρα πολύ σε δεκάδες χιλιάδες ανθρώπους πριν ξεκινήσουν οι εμβολιασμοί και σε δεκάδες εκατομμύρια ανθρώπους από τότε που ξεκινήσανε. Οπότε πραγματικά το ρίσκο είναι πολύ μεγαλύτερο το να μην πάρετε το εμβόλιο παρά το να πάρετε το εμβόλιο. Αυτό είναι το πρώτο. Το δεύτερο είναι αυτές οι επεμβάσεις που κάνουμε, σε τι ρυθμό ασκούν το effect που έχουν. Και αυτό ομολογώ ότι με την άσκηση είναι κάτι το που αρχίζει πολύ γρήγορα και ότι κρατάει μακροχρόνια. Και όχι μόνο φτιάχνει το σώμα μας, φτιάχνει και το κέφι μας, φτιάχνει τον τρόπο που μπορούμε να κοιμηθούμε, τον τρόπο που σκεφτόμαστε. Όλα αυτά είναι συνδεδεμένα. Οπότε κάντε το πρώτο βήμα, γιατί το δεύτερο και το τρίτο βήμα θα έρθουν πολύ πιο εύκολα. Αυτό είναι το πρώτο. Και το δεύτερο είναι ότι για μοριακή επέμβαση, τα διάφορα φάρμακα που παίρνουμε αλλάζουν διάφορα κυκλώματα και μερικές φορές αυτές οι αλλαγές θα ξαναγίνουν όπως ήταν πριν, όταν σταματήσουμε να παίρνουμε το φάρμακο. Άλλες φορές μπορούμε να καθαρίσουμε κάτι το οποίο θα μας δώσει 5-10 χρόνια ζωής χωρίς φάρμακα. Αυτό εξαρτάται από την κάθε ασθένεια και το κάθε φάρμακο και το πώς λειτουργεί. Αλλά υπάρχουν πάρα πολλές ενδείξεις αυτή τη στιγμή που μας βοηθάνε να κρίνουμε και την τωρινή και τη μακροχρόνια αλλαγή που θα γίνει από το κάθε ένα από αυτά τα φάρμακα.
Πολύ ωραία. Σας ευχαριστούμε τόσο θερμά για αυτή την πάρα πολύ εμπνευσμένη ομιλία, για όλα τα μηνύματα που μας δώσατε, για το διαμοιρασμό της έρευνάς σας. Και είναι τιμή μας που σας φιλοξενήσαμε φέτος στο Athens Science Festival. Ευχαριστούμε.
Να είστε καλά και ευχαριστώ για το έργο σας.
Ευχαριστούμε.